Good afternoon, everyone. I think we can get started with this World Café meeting, which is the second in a series that started 2 weeks ago. Today, I'm going to present a number of aspects of the STOP project. I'm Franco Sassi, first of all, I should say. I am a professor of international health policy and economics at Imperial College Business School, and I'm the project coordinator for the project called Science and Technology in Childhood Obesity Policy, which has started 3 years ago when the European Commission decided to make a big investment in tackling childhood obesity, or rather in producing research that would help European governments tackle childhood obesity, and decided to fund two projects, of which STOP is one. The second one is called Co-Create, and was funded at the same time and coordinated by the Norwegian Institute of Public Health. These two projects have joined forces to produce research that is going to change, hopefully, the landscape of childhood obesity in Europe in the next few years. And additionally, we have coordinated with two more projects, one of which has been funded pretty much at the same time as STOP and Co-Create, and that's called PEN, the Policy Evaluation Network, which covers a number of European countries and looks at behaviors like diet and physical activity and how to improve them through government policies and epidemiological surveillance. And with another project that has been funded more recently, which is called Best Remap. It's a European joint action that is focusing on three types of policies in particular to address obesity. One is public procurement of food in public organizations, then reformulation of foods by food manufacturers, and finally the regulation of food advertising to children. So these four projects together are really trying to create a critical mass of research to influence policy making in the areas of obesity, and particularly childhood obesity, in the years to come. And the STOP project has the ambition in particular to work as a bridge between the science around childhood obesity and the policy that governments need to implement, or policies that governments need to implement to address the problem. The World Café today, as I mentioned, is the second in a series. 2 weeks ago, we had the first one, which was led by colleagues in the PEN and Best Remap projects, and in which we explored a number of aspects of the food and physical activity environments in which children make their choices of food and physical activity, and how these environments can be improved to make them less conducive to obesity, including an important aspect of it was the reduction of exposure of children to marketing of unhealthy food products. The World Café 2 weeks ago also explored the way in which the moving database that has been created in the context of the Co-Create project can actually be used as an accountability framework for countries to improve their policies on physical activity in particular. What all of these four projects have in common is that we're really trying to focus on upstream approaches. So we want to change the environments in which children live. We want to change particularly the food and the physical activity environments in which they live their lives through regulation and through other policies in order to be able to reach the children that are most in need for help in terms of behavior change, and these are particularly children in lower socioeconomic status and in vulnerable circumstances, social and economic circumstances. Now, today's World Café is focusing on the causes of childhood obesity. That is one aspect that the STOP project has investigated, especially in detail. So we are focusing on particularly the exposures that the children are subject to even before they're born. So we're looking at the risks that are connected with the prenatal period and sometimes even with the preconception period through the behaviors that the children's parents engage in and the environment in which they live. These early life or pre-birth exposures are increasingly being researched and increasingly being associated with a risk of developing childhood obesity later in childhood. We are also looking in STOP at the built environment, which is going to be another aspect that we're going to address in today's World Café, and particularly the types of exposures that the children are subject to in the environment in which they live, which, as I say, is in common with some of the other projects that we are joining forces with. The idea in STOP is that we can try to identify causal pathways that lead from these exposures to the development of childhood obesity and identify entry points for policy to break those causal pathways and make sure that those exposures do not eventually lead to childhood obesity or to remove those exposures altogether when that is possible. And we want to do this in view of providing recommendations to policymakers for improving policies in their own countries. Now, before I give the floor to the first speaker, I would like to remind everyone who is on the call about some housekeeping rules. First of all, questions will be asked via the Q&A box, so please do that. Put your questions into the Q&A box so that they can be picked up by the moderators and asked to the speakers. Attendees are welcome to make comments or reflections in the chat box, so you're quite welcome to add your remarks there. And the webinar will be recorded and shared only with attendees, so hopefully you will be in agreement with this. Now, in terms of the agenda of the meeting, if we can move the slide to that, the agenda involves 3 presentations that will be given by Joseph Tour, Oliver Robinson, and Elisa Pineda that will explore the aspects that I mentioned a minute ago. And then we will have a panel discussion, a 15-minute panel discussion that will try to reflect on the lessons from those 3 presentations before closing remarks in the final part of the meeting. You will be able to ask questions, as I say, in the Q&A box throughout the presentations so that we can pick them up at the end. So without further ado, I'll introduce our first speaker today. That's Joseph Tour, who is head of a research center called Cyber OBN, which is a research center focusing on obesity as part of the Spanish National Institute of Health, Carlos III. He's based in the Balearic Islands, so we are all very envious at the moment. We wish we were all on a beach. But Pep has been doing a very large amount of research within STOP and elsewhere on the determinants of childhood obesity. So I'll hand it over to him for his presentation.
Thank you so much, Franco, for your kind words. And let me thank the organizers to invite me to participate in this World Café. As Franco said, and said very well, I tried to introduce in the preliminary influences that determine the childhood obesity, speaking about the prenatal and early life influences. Next, please. As you can see in this slide, we are influences to arrive to achieve the obese status by a lot of factors that using a technical word that the researchers use, that is called the determinants. So there are a lot of determinants that influences on us to arrive this obesity. One of these determinants or factors is the characteristic of the microbiota. The microbiota is the bacteria that lives into our body, in the different parts of our body, and most of them are living in our gut, in our intestine. So as you can see in the slide, there are lots of bacteria living into our gut, and according to this, is establish a sort of interrelationship between our metabolism, our immune state, and the population of this or kinds of this microbiota. And so that when we eat a sort of unhealthy diet, plenty of fats or saturated fatty acids, simple sugar, etc., these nutrients we ate uh, came into our metabolic routes, and the final is that we accumulate a lot of fats into the special cell dedicated to accumulate fat. It's called adipocytes, or the white adipose tissue. According to this adipocyte, this cell specialized to accumulate fat, is submitted to a sort of an injury, sort of a strain, and then the a consequence of this strain appears a lot of molecules that acts on these adipocytes, the same adipocytes but also to a systemic way. In other words, the fact that we accumulate fat into adipocytes uh, produce a lot of inflammation, local inflammation in the adipocytes, but also these inflammation molecules or cytokines travel by means of the blood vessels, arriving to a lot of organs, important organs like liver, like the pancreas, like kidney, etc., and produce a systemic inflammation. So we change the immune response, we change the inflammation and the function of several important organs, and also we change the metabolism in that way, appear a lot of metabolic molecules in our blood that could arrive to produce so important diseases like cardiovascular events, ictus, hyperlipidemia, or diabetes mellitus, etc. But also this um, this accumulation of fats in our metabolic routes also arrive to the gut, modifying modifying the population, or the kind of population of bacteria or microbiota into the gut. Into the gut there lives a lot of different bacteria. One could be healthy and another unhealthy, and depending of the diet we used, this population could change from healthy microbiota to unhealthy. In this case, if we are working to be obese taking this unhealthy diet, so the unhealthy microbiota grows and the healthy microbiota decrease. So this increasing in unhealthy microbiota also produce an additional fatty acids that modify the characteristics of the cell membrane of the intestinal cells called enterocytes, increasing the absorption of more and more fats and triglycerides, etc., increasing the obesity state. So the microbiota in monostatus and metabolic is called the MIM. It seems like a name of candies, M&M. So microbiome, immune, and metabolic axis. That is very well demonstrated the relationship with the adult obesity, but not so well in the childhood. At least the knowledge we have it mainly came from the animal studies. The next one, please. So this axis, microbiome, immuno, and metabolic axis, could be influenced by a lot of factors. Most or important factors are are represented in this slide. That you can see, for example, the maternal diet. We spoke about this. If you take an unhealthy diet, like high protein, high fat, high calories, inducing the change in the microbiota from healthy to unhealthy, that is called technically as dysbiosis, producing inflammation. The risk of obesity is increased, as I told you. Also, the maternal health, the state of health in the mother could change. Not only the mother, but also the future characteristics of the fetus or the future children. And what sort of health? For example, if the mother is obese or is diabetic or any others, because this status, this disease in the or unhealthy in the mother could change the expression of the genes to be genes that express proteins, not gluten, also to pass several metabolites to the fetus and increase the risk of the future child would be obese. But also, depending on the antibiotic use that is taken by the mother, could conditionate the final kind of microbiota, could increase or decrease a special kind of bacteria, changing from a healthy to healthy microbiota. But also, it depends on the birth method. For example, if the birth method is normal, as classical, then the maternal via or via vagina, and so as the vagina also has bacteria inside, so the fetus, the newborn, when passed through the vagina, was contaminated with bacteria, but this bacteria would be beneficial for them. However, if the method is the cesarean, so the bacteria that will contaminate the newborn will be the bacteria from skin, and usually this contamination uh, goes to a microbiota not so healthy than those that the newborn could take from the vagina. And the last factor to comment now is the feeding methods that the newborn receives. So if this newborn was fed by human milk, breastfeeding, usual breastfeeding, this breastfeeding of this human milk from the mother is plenty of oligosaccharides. Oligosaccharides, in a method to understand everybody, is the food, a special food for healthy microbiota. However, if the newborn was fed by a formula, but the industrial milk, in other words, this formula usually have not oligosaccharides, and so the healthy bacteria will not be promoted. Next one. So after that, what sort of questions are asked to be solved? For example, when and at what stage depends of what factors are better to determine the kind of microbiota. For example, during the fetal life, the most important factors are the maternal diet, the gestational age, and the gestational weight. When in the newborn, so the lactating children, the most important factors are delivery mode, feeding, the weaning diet or the diet that they receive after weaning, the use of antibiotics, the metabolic state and maternal diseases, and when we're told that in the early childhood, they are better from the best, the most important factor of diet, they receive environmental and hygiene factors. In other words, all these factors conditioning the dysbiosis, that is the most important factor that we must avoid. Another factor that could condition the future childhood obesity, the role of birth weight, for example. It is well demonstrated that a low body weight and small gestation age children have higher risk of diabetes mellitus type 2 and insulin resistance. this relationship between both the low weight, small gestation age and this risk is not very well known. However, most of the researchers think that it is associated to impaired glucose metabolism, and so that the obesity is very close. As the prevalence of preterm babies in the last times is very high, and so it is a vulnerable population, and so this is a factor to be controlled. And finally, the use of antibiotics in the fetal life. There is not a clear association between antibiotics that affect the mother and child obesity, and just it could be a high risk during the second trimester of gestation, and also if the mother takes more than 3 repeated courses. With the newborns and the lactating children and the toddlers, just they could show some increased risk of obesity after 4 continuous courses of antibiotic therapy, or after they were 6 more than 6 months old, they increase or decrease the risk of obesity, increase it when the mothers are normal weight, or decrease if the mothers are obese. And we were seeing this decrease because in the obese mothers, the unhealthy microbiota is higher, and so the antibiotics will be beneficial. And whereas in the normal weight mothers, the microbiota are mostly healthy, and so the antibiotics decrease this healthy microbiota, and so the obesity could be increased. Next slide, please. All these last results we're working in the 3 different reviews that will be published in a future special issue on obesity reviews dedicated to determinants on childhood obesity. And in the top of this slide, you can see the title of the reviews and the authors responsible of these results. Thank you so much for your attention.
Thank you very much, Pepe. That was a very interesting presentation. I see a question in the chat. Is there an impact of antibiotic use while breastfeeding on childhood obesity outcomes?
As much as I know, usually not. However, each person is different, and so my best counsel is if you before to take antibiotics during the breastfeeding, it is better to consult with your pediatrician.
Thank you, Pep. I'm conscious of time. Just a very quick question from me. Is there any epidemiological evidence linking? I mean, you've been talking about birth methods and the potentially increased risk of childhood obesity with cesarean sections. Is there any epidemiological evidence that where there are more cesarean sections, childhood obesity is more prevalent?
It's difficult to say. I had a lot of different opinions about this. As much I can read about it, establish a difference between the different methods. However, by protocol in the hospitals, they apply a preventive how to say therapy. It depends. The present protocol in the hospital is applied, but however, is not well demonstrated until now.
Thank you, Pep. That's really helpful. I will hand the floor to Oliver now. Oliver Robinson, who is a lecturer at Imperial College London at the School of Public Health, funded by the Medical Research Council, and has been the scientific coordinator of a large European project before called Helix that was focusing on children in various cohorts around Europe. Over to you, Oliver.
Thank you, Franco, and thank you, the organisers, for this great webinar and inviting me. So I'm going to talk about epigenetic and molecular determinants of obesity. Much of this will be from 2 systematic reviews we prepared to get an overview of the state of the evidence and the gaps, and these will be published in the same special issue that Pep mentioned earlier. And also I'll talk about some ongoing research coming out of these reviews. So if we go to the next slide. So we know, of course, that the genome has a strong effect on childhood obesity. Heritability estimates from twin studies put this at anything from 30% to 80% additive effects of genes on childhood obesity. And what's interesting is that these estimates, the higher estimates of heritability, are found in families who have more obesogenic environments or are more socially deprived, suggesting, of course, this strong interaction with the environment of how these genes are expressed. So if we look at the next slide, how genes are expressed to affect childhood obesity risk, there are several levels of biological organization that we're interested in. So we have the epigenome, which is the molecular machinery that controls which genes are switched on or off. And you can see in this strand of DNA in the epigenome, we're interested for a lot of studies, we have looked at methyl groups, which are small methyl groups attached to the DNA, which control its expression, termed DNA methylation. And these control how DNA is transcribed into RNA and how RNA is translated to proteins. And then it's the proteins that control the metabolism. And from this, we can assess the profile of small molecules in blood, the metabolites, as a pathway from the genome to childhood obesity. But if we look at the next slide, of course, we're very interested, particularly in the epigenome and the metabolome, because as well as the genome, they're very strongly influenced by the environment, with DNA methylation marks capturing long-term changes, which can have quite profound effects during fetal development in terms of metabolic profiling, so could act as a determinant of childhood obesity risk. And the metabolome, of course, these small molecules are also strongly determined by the environment. Much of the profile is determined by the food we eat. So it's a very important mediator between the environment and childhood obesity. So if we look at the first systematic review on the next slide, we did a systematic review of all the literature on epigenetic signatures of childhood obesity. So we looked at children up to 18 years, epigenetics of various types, comparing overweight or obese to non-overweight children, or continuous variation in parameters such as BMI. In the end, we had 94 studies focusing on DNA methylation. And if we look at the table at the top, we can see in the general overview, 15 studies looked at imprinted regions, and these are very interesting regions because they're maternally imprinted. So they're another way that the maternal prenatal environment can get passed down to the children. And 2 regions were looked at by more than one study, but we see mixed evidence for these of associated and non-associated. But interestingly, they're all the associated studies are in the same direction. Other studies, a majority of the study looked at candidate genes, so genes that we know are involved in obesity from GWAS studies, looking at their methylation levels. 2 genes came up in more than one study, the LEP gene in 9 studies, and the HIF3A study in 4 studies. So we have quite mixed evidence for LEP, but of the 4 studies, we did see a positive association with HIF3A, including a study looking at cord blood methylation and later obesity risk, which is quite interesting. Though I think it's quite telling, of the 19 EWAS studies we looked at, while 15 did find an association, none of these candidate genes were replicated, and only 3 sites are replicated by more than one study. So there are 3 genes that were methylation was reported in 2 studies each. So I think, to summarize, several studies showed that evidence that childhood obesity is linked to epigenetic changes. However, most of these have not yet been replicated despite, I think, 10 years or so of research. And few studies have actually investigated causal direction. So is this a cause of obesity, or could these actually act as a determinant of obesity, which is what we're really interested in? And these studies did seem to suggest that most of these methylation changes are a consequence of obesity. So I think there are still several gaps and limitations in the literature, particularly in population in terms of the size. We need very big studies for this, and more homogeneity across studies in how they choose their covariate adjustments in the tissues, for instance. So that's the state of the epigenetic evidence. If we look at the molecular metabolomic evidence, we performed a similar process, at this time looking at studies that looked at metabolomics. This is techniques that profile at least 10 molecules in urine, saliva, or blood samples. So far, we found 41 studies, 27, the vast majority were cross-sectional studies in blood. Over 200 different metabolites were associated, but in this graph, you can see that the metabolites associated in more than one study, and a consistent profile is emerging, particularly steroids at the top, various lipids and fatty acids, acylcarnitines, particularly propionyl carnitine, showed consistent increases, but the strongest profile was for the amino acids, the branched chain amino acids, valine and leucine, which showed many studies with an increase associated with obesity. But these were mainly cross-sectional studies. There were only 5 studies that had a longitudinal design where we could look at determinants. And while there was some evidence for some of these molecules having an effect on later obesity risk, in particular leucine, these studies were also generally too small to draw firm conclusions, considering the number of molecules that were looked at in each study. So to address this gap, if we go to the next slide, we performed, if we look on the left, the cord blood metabolic signatures predicted of childhood overweight. We performed an MWAS study using untargeted mass spectrometry in 400 newborns. So this is the largest study to date. You can see in these red dots in the metabolome-wide association study plot, there are a number of features that were associated with obesity at 6 years. These could be grouped into 8 metabolites. And actually, if you look at the ROC curve and you look at the blue line in the ROC curve, you can see that addition of these metabolites to traditional risk factors shows a marked improvement in predictive ability. So there seems to be some utility in screening cord blood to stratify children by obesity risk. And what's interesting is the most strongly associated were the branched chain amino acids valine and leucine. And here lower levels were associated with later overweight. And finally, we looked at ultra-processed food, which we know is an important component of the obesogenic environment in Ausbach children, a British cohort. So we looked at NMR profiles in over 4,000 children. And in this same population, we know that ultra-processed food increases the risk of later development of adiposity, with differences emerging by about 13 years. And if we look at the red bars in this forest plot, it shows the profile at 7 years. Again, we see changes, a shift in lipoprotein profiles, lower levels of the better fatty acids, omega-3, and polyunsaturated, but also, again, we see this lower profile of branched chain amino acids, valine and leucine. So I don't know if you can see this, but there does seem to be this paradox that we see consistent evidence of higher levels of branched chain amino acids associated with obesity itself, which is probably a result of changes to overall metabolism associated with this metabolic disorder. But actually, many intervention studies and feeding trials have shown that actual higher levels of these branched chain amino acids has beneficial effects for weight and appetite control. So it's quite a complicated picture. So if we go to the next slide, I think
Oliver, I will ask you to wrap up if you can, so that we can ask you a couple of questions.
Yes, of course. So I just wanted to make the point that while we were looking for determinants, we have to consider that of all these molecular features will have a strong effect from childhood obesity itself, which makes things, as always, quite complicated. And these are the acknowledgements of my collaborators on the various different studies. Thank you very much.
Thank you, Oliver. Thank you. That's great. And sorry if we, unfortunately, are very tight on time. I wanted to have an opportunity for you to answer a couple of questions. One from Eleni. Among overweight and obese children, can we identify some that are metabolically healthy and others that are metabolically unhealthy? Is that possible?
Yes, of course. I mean, traditionally we do it by measuring the lipids and their glycemic control using traditional clinical risk factors. There's very nice research that people have a metabolic profile, train it with much deeper metabolic measurements than these clinical markers to profile what is the profile of obesity. And there is a difference, you know, even in healthy people, those that have the metabolic profile in normal weight children, they can have this obese metabolic profile, and this has similar risk of later diseases, BMI measured by overweight itself. So certainly this is one application of metabolomics, and I think this is very useful. As you saw from that profile, all of these different metabolites have a role to play in other diseases.
Thank you, Oliver. Just a last quick question from Veronica. Are there differences between boys and girls in their epigenetic and metabolomic profiles and impacts on childhood obesity?
That wasn't an aim of the systematic reviews. So it wasn't something that we actually pulled out, but I can't remember in the reviews any of the papers actually delving into that. Certainly in the research I showed, these profiles of later obesity risk were similar in boys and girls. So that there isn't any evidence so far of a difference, though I didn't look too closely in the systematic reviews.
Thank you, Oliver. That was an excellent presentation. I'll move quickly forward in the program and hand the floor to Elisa Pineda, who is a research associate in the Center for Health Economics and Policy Innovation at Imperial College Business School, and is going to talk us about the environment, particularly the school environment in which children live. Over to you, Elisa.
Thank you, Franco, and good afternoon to everyone. Yes, so I'll talk to you about the systematic literature review and meta-analysis we did on interventions of the food environment and identifying which ones are effective in terms of preventing childhood obesity. So looking at the next slide, we see that schools are great settings in which to carry out interventions for the prevention of childhood obesity, because these are the places where children spend most of their time. And also, according to the literature, more than a third of their meals and snacks are eaten at school. So this is a very good place to begin with in terms of looking at strategies, so putting in practice the evidence. So on the next slide, we look at the methods. So in the next slide, we have the aim of this systematic review and meta-analysis was to assess the different types of interventions that were carried out at the school level, specifically looking at how effective they were at improving either dietary intake or preventing actual childhood obesity. And therefore, we looked at interventions that were carried out either within the schools or around the school setting. So physical or structural settings. So in terms of the aspects that we focus were school children of maximum 19 years of age, according to the WHO categorization. And then in terms of interventions, we focused on either interventions that assessed how to improve dietary intake, food provision, or modifications also of the infrastructure, modifications of meals, or any aspect that had to do with the physical setting of the food environment, such as food choice architecture. In terms of outcomes, we focused on all types of outcomes related to obesity, dietary intake, or those related to food purchasing as well. And finally, what we focused on in terms of the methods were the estimates from each type of study that we looked at. We gathered all the evidence and we pulled it together to carry out a random effect meta-analysis. And we stratified these results by their different outcomes and methods, and we focused specifically on the most common outcomes, which were BMI set score for children, food, and vegetable intake. These three were the most common presented by the literature. And then we did a risk of bias assessment. So in the next slide, we have the what is the food environment specifically in schools. So we identified either interventions focused on spaces, infrastructure, or conditions of the food environment. So this could have been the presence of a kiosk, a canteen, a food vendor, or a vending machine, for example. But we also took into consideration the provision of food and the quality or the nutritional quality of the food that was provided, whether modifications had been carried out. And lastly, we also considered either promotion of food, such as marketing or advertisements, or any type of information that was made available to the children. And lastly, pricing of food. So in the next slide, we can observe the different components that make up the school food environment that we considered for this study. So either infrastructure, modification of meal plans, or reformulation of the meals, also staff training, food display, food accessibility, for example, within a canteen or vending machine. It could also be food provision. This could usually be free, or it could have a cost. It could be in the form of a snack, breakfast, or lunch, which were the most common. And also it could be different types of food availability, for example, fruits and vegetables, and also how they're presented in terms of color and sizes. It all seems to make a difference in terms of food choice, specifically for children in schools. So what we found in the next slide is that we identified 100 studies that were from 27 different countries. From these studies, 51% demonstrated that there was a decrease in BMI from the interventions that were carried out from these studies, or healthy choices were increased as well. And more specifically, looking at the health outcomes that we looked at, which were BMI set score, we identified after carrying out a meta-analysis that these studies, when pulled together, show us that there could be a 2% decrease of obesity prevalence when undertaking modifications of the school food environment to improve food choice and therefore prevent childhood obesity. Also, when looking at food intake, we found that those specific interventions that either provided food or made modifications in the way that food was provided, those were, when pulled together, the studies showed that there was a 15-gram increase of food per day consumption. However, it was interesting to find that significantly speaking, none of the interventions showed effective ways of increasing vegetable intake in a sustainable way. So that was one of the findings that we assessed or identified when pulling together all the studies. And one also important aspect was that children with lower socioeconomic position backgrounds were more likely to have a higher risk of obesity. However, very few studies address social inequalities when either designing the interventions or also when assessing the impact of the intervention at different levels of income or education or socioeconomic position. So what is taking lessons from these findings, we can say from the successful studies that it seemed that those interventions that provided very clear guidelines on what types of food should be permitted or banned in canteens were more likely to be successful in either increasing healthy dietary intake or decreasing obesity. Also, those that focused on regulating portion sizes, also those that made through strategies of food placement, those were more effective in making healthy meals and snacks more visible and more attractive for easy access. Also, those which focused on increasing the taste and healthiness and attractability of the meals by using professional chefs also seem to have an impact. Also, of course, modifications of the meals that were provided either for reducing sugar consumption or providing water instead of sugary drinks. Also, being very inclusive in terms of providing very clear, precise guidelines to staff, teachers, parents, and so that everyone had the right information and was implemented effectively in terms of improvement dietary access within the canteens or the physical environments that provided food around schools. And lastly, those studies that address social inequalities seem to be more effective as well in creating a greater impact that was sustainable. And also, of course, the studies that monitored the implementation of the interventions through time were more likely to sustain the effects or the associations of healthier dietary intake or lower BMI and sustain that through time. So in our next slide, just want to thank you and happy to answer any questions or comments. Thanks.
Thank you very much, Elisa. A very clear presentation. I see one question in the chat. Can you comment on procurement policies in schools? Do you have any quick comments on that, Elisa?
In terms of, so the interventions that were included here were only of those that provided food directly to the children within the school environment. So that could have been breakfast, snacks, or lunch. Those were the three types of food provision that were identified, more than food procurement. We only focused on food provision in terms of a potential intervention. The other types of interventions that were considered were modifications of the infrastructure. That could have been, for example, the inclusion of a salad buffet bar or a vending machine or an elimination of vending machines or modifications of certain aspects of how food was displayed as well. So those were the elements of the interventions that we focused on for this study.
A couple more questions. I'm conscious of time, but there's a question about social inequalities. How did the studies address this dimension, Elisa?
Yeah, so there were specific studies, especially in the US, that addressed meals exclusively for lower income populations or minorities, and they adapted them to either their culture or the specific needs that that population had, and were very precise in how the foods were presented to them in terms of promotion, in terms of availability, and free access. Whereas other interventions which didn't make these healthy foods available in a sustainable way and in a lower cost or in a free form were not successful in the long term. So these were some elements that we found in terms of social inequalities.
Last very quick question for you, Elisa. Did you find any evidence in that literature about the value of using things like cartoon characters or testimonials in these interventions?
I am aware that there are many studies that have focused specifically on the use of cartoons or elements like that. We did not, however, include those very specifically here, only those which focus very broadly on marketing or promotion, which we did identify there was a benefit in increasing healthy food choices, but it was not specifically, we didn't address specifically which form of promotion it was, whether they used images, or it could have also been, we grouped it together, if it could have been promotions of 2 for 1 in terms of vegetables or sizes, but not specifically cartoons. But I am aware that there are studies that have focused on that specifically.
Thank you, Elisa. Just to close off on the procurement question, I mean, Elisa's review and presentation has shown very clearly that there is a value in limiting access to unhealthy foods within school environments. So there is at least indirect evidence that, you know, food procurement that selects the foods that are purchased by school canteens to avoid less healthy foods would clearly, you know, be beneficial. Of course, we don't know what the spillover effects might be on foods that children purchase or consume outside of the school environment. That is clearly a separate question that needs to be addressed as well. So thank you, Elisa. I'm conscious that we are running over time and we'll have to squeeze the panel discussion, but we have 3 very interesting speakers. So I would like to hand over to our first speaker in the panel discussion, who is Natalie Savona, based at the London School of Hygiene and Tropical Medicine, who is going to give us an interesting perspective. Natalie works on the Co-Create project, and which focuses on adolescents in particular. And I think it would be very interesting to hear from Natalie what the view of adolescents is on what the drivers of childhood obesity may be in European countries. So over to you, Natalie.
Thanks, Franco, and thanks everyone for having me on the panel. So I'm conscious of time, so I will dive straight in. What you can see here is a system map that we created as part of Co-Create, so working with 16 to 18-year-olds, and it depicts what they see as the drivers of obesity through physical activity and diet. And I realize obviously you can't see all the details on that, but I'll just highlight, if I may, a couple of what we think were the particularly interesting findings. So on the right-hand side, the purple boxes that you can see are very much under the theme of what we called emotional, mental pressure and time. So sort of stress, how young people are experiencing stress. And then on the other hand, on the left-hand side, the sort of pinky fuchsia boxes are ones that are connected to economic and commercial influences. And then there are others, the green is physical activity, and I won't go into all the different colors because I want to come back and draw out a few what we think are particularly interesting factors here. So another word for a system map, as I'm sure most of you will know, is a causal loop diagram. And so what we really were able to get to explore with these young people was, if you like, the causes of the causes. A lot of work is done on healthy diets, unhealthy diets, physical activity, and what drives it or not. But what we felt we were able to uncover with this method is a bit more of their views on the causes of the causes. And obviously, if we are going to be putting policies into place, then it's worth considering, if you like, the targets of those policies, the targets of those interventions, and what they see as the drivers of the problem at hand, in this case, body weight. And what we drew out of this causal loop diagram, which is an amalgamation of 20 separate causal loop diagrams that we created in individual sessions across 5 countries, is that the issues that really came out for young people were this idea of mental health driving the way they perceive their bodies and the way they might eat or you know, not eat particularly healthily. And we brought out some feedback loops from these diagrams that in a causal loop diagram are often very fruitful places for intervention. And I'll just focus on one of those, which is very much that they were saying that if you've got stress going on in any form, it affects your mental health, which might affect what you're eating and your calorie intake. And that in turn might affect your body weight, which goes on to affect your self-esteem about your body. So there is a lot more I could say, as I'm sure you can imagine, but I'm conscious that we should move on and hear from the next person. But of course, I can answer some questions. All this to say that I think there's some really interesting ideas that we can work with to come up with potential levers for change in this scenario. Thank you.
Thank you, Natalie. And talking about levers for change, both our next speakers, Margot Neveu and Paolo Binese, will talk about policy solutions. Margot is a senior policy manager at the World Obesity Federation, and she's going to talk about the roots framework in particular that the World Obesity Federation is launching.
Thank you very much, Franco. And so I will try to be brief and share some resources in the chat. But so for those of you who don't know, so on World Obesity Day 2020, so March 4th, 2020, the global obesity community came together to really actively advocate and acknowledge the complexity of obesity to develop what we now call the Roots framework. And so the Roots framework really aims to provide some suggestions for obesity policy and advocacy that can be adapted to different national settings, with a series of recommendations that are aimed to provide some ideas and some inspirations that can be adapted in different settings to really address the root causes of obesity. Another thing that we saw is that, as most of you are probably aware, COVID-19 really disrupted health and food systems around the world, but also obesity very early on emerged as an independent risk factor for worse outcomes in cases of illness due to COVID-19. And so highlighting the urgency of action, we then came back to refine the Roots framework at our Global Obesity Forum in 2020 to really incorporate the impact of COVID-19. And so what does Roots stand for? So we call to recognize that obesity is a disease in its own right, but also a risk factor for a number of other conditions, including significantly worsening outcomes of COVID-19 infection. We also asked for obesity monitoring and surveillance system to be enhanced to really strengthen effective strategies for both preventing and treating obesity. We also called for obesity prevention strategies to be developed, tested, and implemented across the entire life course. So that means from preconception through childhood and into older age. We call for treatment of obesity to really include behavioral, pharmacological, digital, nutritional, physical activity, and surgical-based solutions. And finally, we call for, and that goes back to what Natalie was just presenting on, to adopt a systems-based approach that really can be applied from the prevention and to the treatment of obesity. And so in terms of recovery from COVID-19, we really call for action across the health, the food, transport, and all of the other systems. And so what we have seen over the past year is that COVID-19 truly shows that there is an urgency to act to really address the underlying obesity, but there is also an inspiration for political and collective action. And we really have an opportunity to fund new, innovative, and bold interventions to address obesity globally. And I will share some more information about the Roots framework in the chat very shortly.
Thank you very much, Margot. I should also point out, I mean, we're going to be running over for a few minutes, and I apologize to our audience for this, but hopefully you can stay on the call for a little bit longer. There are a few questions in the Q&A box for other speakers, for Oliver and Lisa, as well as Natalie. So if you want to answer them by typing your response there, you're welcome to do so. Paolo Binese is a chair of environmental epidemiology at Imperial College and a leading researcher in the environmental determinants of health. So I'll hand it over to him. His talk is a perfect conclusion for this webinar, as he's going to highlight some entry points for policy, starting from the evidence that the presenters have displayed in their presentation. So over to you, Paolo.
Thank you, Franco. Just a couple of points I want to stress. First of all, consistently with what Margot and Nathalie said, I want to stress the idea of syndemic that came up recently thanks to an editorial by Richard Horton in The Lancet. But in fact, the idea was coined in 1994 by Mary Singer to stress the fact that very often diseases or traits or conditions go together, and also socioeconomic inequalities play an important role. So we have seen it for COVID-19 with the role played by socioeconomic inequalities, but also the susceptibility background represented by obesity, hypertension, and other conditions. Well, I think that also childhood obesity could be looked at in this perspective of the syndemic, which is helpful for several reasons. So, for example, Nathalie mentioned stress and mental health, which goes together with predisposition, at least to childhood obesity. But there is also a loop where obesity itself leads to further stress and mental health. So my first point is that instead of addressing each disease or each condition separately, we should invest more into looking at combinations, interactions among diseases, and also the role of social inequalities. My second point is related to another problem that is affecting humankind that requires immediate action, which is climate change. And I refer to that because we have a lot of opportunities to address also non-communicable diseases starting in childhood, including obesity, together with our action against climate change. I refer to the Next Generation Plan, for example, Green New Deal, and so on. Why I'm saying that? Because determinants of childhood obesity include factors that also affect climate change. I refer to ultra-processed food that was mentioned by Oliver. I refer to sedentary life, lack of physical activity. Both are determinants of childhood obesity, but in the meantime, they are related to environmental changes that predispose the planet to further changes and disruption, including climate change. For example, ultra-processed food, in addition to encouraging overeating, also drives land use changes. In general, the current food chain, food industry, including the primary commodities, but also processing, packaging, distribution, has a dual role in promoting obesity on one side and leading to environmental changes that include climate change. So I would like to mention also that certain activities that are associated with sedentary life, like TV watching by children, which seems to be to occupy 3 hours per day in the United States, which means about 200 watts in terms of electricity. Also, TV watching, overuse of electronic games, and so on, are a dual risk factor for both obesity and planetary change. I stop there, but I think that it is plenty of opportunities for interaction with policies around the environment and climate change. Thank you.
Thank you, Paolo. I'm really grateful to all 3 speakers in the panel discussion for being able to provide their comments in a very succinct way, which allows us, albeit with a few minutes delay, to conclude this webinar, I just wanted to recap the key messages that there are major determinants of childhood obesity at play in early childhood and even before birth. And one of the ways in which we are now able to get evidence on these childhood obesity determinants is by looking at biomarkers, a range of biomarkers that have been illustrated in some of the presentations today. The key question, of course, that we are still in the process of clarifying the causal pathways, and at this stage we're not entirely sure that some of these biomarkers are caused by obesity or whether they are part of the causal pathways to obesity. So I think elucidating those causal pathways is really a key step in understanding how to tackle childhood obesity. But all of us, not just in the STOP project, but also in the other projects that we are coordinating with, need to make an effort to act upon this evidence and translate that into policy action and effective interventions that can change the course of childhood obesity. I will give the final word to Sally, who has prepared a nice live illustration for this webinar, and that is, I think, a perfect way to end the webinar. Sally, would you like to comment on that briefly?
Hi, everybody. Lovely to meet you. Thanks for having me for your session. I hope that I've captured at least the high level of the topics that you covered today. Yeah, I don't feel qualified, but hopefully visually, if you want to have, I think Claudia will send around this scribing to everybody after the call, so you can have a look at it at your leisure. But thank you very much for having me today.
Yeah, you've been great in capturing in a nutshell the discussion we had today, and we'll post this on our website. Just to remember everyone, the website is www.stopchildobesity.eu, and you're welcome to visit that, and you will find contact details for everyone. You've got the websites of all 4 projects here. You're welcome to visit those, and you will find contact details for all the research teams in case you want to follow up on any of the points that were discussed today. Thank you again, everyone, for your participation. I see the very healthy participation. We have touched the high of about 200 participants during the webinar. Now we still have 150, so I'm really grateful for everyone who stayed on the line until the end. Thank you very much. And Claudia or Margot, would you like to remind everyone when the next World Café will be?
Sure. The next World Café will reconvene in September. And so we'll be circulating specific dates a little bit later in the summer. And just to also specify that the recording of this meeting will be made available in the next couple of days, and it will be circulated to all of the attendees.
Thank you very much, Margot, and bye-bye, everyone.